Humoral immunity kills microbial invaders and toxins that are in the blood or in tissue fluids. It involves T lymphocytes and B lymphocytes, or B cells that develop in the bone marrow of animals. The B cells produce Y-shaped proteins called antibodies. Antibodies bind to specific antigens and mark them as an invader to be eliminated. B cell receptors specifically bind free antigens. When you look in the picture, when that antigen binds to the BCR, that marks a foreign substance that the immune system needs to react to and respond and kill. Central tolerance is important before the lymphocytes mature because it eliminates immature T and B cells found to recognize self molecules, whereas peripheral tolerance is important after lymphocytes mature because it prevents T and B cells that were not eliminated during central tolerance from reacting against self or other harmless molecules.
T lymphocytes, or T cells, are a type of lymphocytes that matures in the thymus. There are three different kinds. First, there's cytotoxic cells, and these are responsible for inducing apoptosis in cells that are infected with viruses or are otherwise corrupt. Next, there's helper T cells, and these are responsible for directing and assisting in many immune responses. Lastly, there are regulatory T cells, and these help control the immune response. Cell-mediated immunity, or also known as cellular immunity, is immunity that involves a T cell response. First, there's dendritic cells. These are antigen-presenting cells that play an essential role in the activation of T cells. Next, there's cytotoxic cells. The process of this is once a T cell is activated, it proliferates and differentiates to become an effector form that's called a TC cell. The TC cell then search for a certain infected cell that they can recognize, and if they find one, they undergo apoptosis, or cell death. Next, there's helper T cells. Once the helper T cell is activated, it proliferates. Some of the clones differentiate to become effector forms that are called TH cells. The TH cells are responsible for delivering cytokines to certain macrophages and B cells. Another form is called memory helper TC cells, and they respond quickly to reoccurring antigens. Lastly, there's regulatory cells. Regulatory cells are important, and they stop the immune system from overreacting and responding to harmless substances.
The nature of antibodies. Antibodies are known as serological properties attributable to an antigenic stimulus, or in more simple terms, they basically are proteins that protect our body from allowing unwanted substances to enter inside of it. They are formed in the plasma by filtration through capillary endothelium, which has absorbed antigen. And then within antibodies, there's going to be an antigenic stimulus, which has results, which is as follows. The antibodies may be formed, which are the precise mirrors of their antigens. Antibodies may arise, which are not the exact counterpart of any particular antigen, or the antigenic stimulus may lead to a complex humoral changes, which are not identifiable as antibodies.
The clonal selection theory refers to the immune system and its adaptive nature to respond to a seemingly unlimited range of antigens by using pre-existing populations of lymphocytes, each one pre-programmed to recognize a specific antigen. Antigen describes the toxins that are introduced to the body. When the immune system encounters a given antigen, the lymphocytes can recognize it and then multiply and attach to the antigens, thus fighting them. Antigens determine which lymphocytes multiply. This involves both T cells and B cells. The first step of this process is lymphocyte development. This involves immature B cells, which are B cell receptors that are randomly generated, as well as naive B cells, which are programmed to recognize specific antigens. Step 2 is activation. This is when activated B cells are bound to antigen X, and then they can proliferate. Step 3 is proliferation or differentiation. This is when plasma cells secrete large amounts of antibodies and bind to antigen X. Also, memory B cells can recognize antigen X, which helps attach more lymphocytes. Step 4 is the effector action. This is when antibodies neutralize invaders and tag them for destruction. Thus, expansion of lymphocytes can only occur when they recognize specific antigens.
Lymphocyte development begins in the fetus. B cells are made in the bone marrow, and T cells are made in the thymus, and a large number of new lymphocytes are made in the tissues. As we age into adulthood, our T cell numbers are maintained through the division of mature T cells outside of the lymphoid organs, and our B cells continue to be made in the bone marrow. Then, lymphocytes become specific towards an antigen through the process of gene rearrangement. In B cells, DNA sequences that encode for the immunoglobulins are adjusted to be antigen specific, and in T cells, T cell receptors are made of gene segments to become antigen specific. After gene rearrangement, the survival or death of a lymphocyte is decided through a process of positive and negative selection. When a lymphocyte has receptors that interact weakly with its self-antigens or bind self-antigens in a certain way, it is destined to survive. When a lymphocyte has receptors that bind strongly with self-antigens or interact strongly with self-antigens, it is destined for death. Finally, the lymphocytes that survived are then sent to the lymphoid tissues to finish maturing. The number of mature lymphocytes is very small compared to the amount that was originally generated. These mature lymphocytes are then able to produce a wide variety of immune responses.
Human immunity beautifully aligns with the Christian worldview. The human immune system serves as a reflection of God's intricate design and care for his creation. As Romans 12:4-5 illustrates, just as our physical bodies possess many interconnected parts, each with their unique function, so too does the body of Christ encompass diverse individuals, each contributing to the unified whole. In a similar manner, the immune system consists of a vast array of cells, antibodies, and organs, all working in harmony to defend the body against harmful pathogens. This unity amidst diversity echoes the Christian principle of interconnectedness and mutual dependence, where believers are called to support and uplift one another in their collective journey of faith. Also, the resilience and adaptability exhibited by the immune system mirrors the steadfastness and grace of God's protection in the face of adversity. In times of illness or challenge, the immune system demonstrates its remarkable ability to adapt and overcome. Just as the immune system tirelessly combats invaders to maintain the body's health and integrity, believers are encouraged to persevere in their spiritual journey, trusting in God's guidance and provision. Thus, the human immune system not only serves as a testament to God's wisdom and sovereignty, but also serves as a profound reminder of the enduring strength and unity found within the Christian community.
Nursing application. A nurse's job is to provide and coordinate patient care and educate patients and the public about various health conditions. To do this well, a nurse needs to have knowledge about how the immune system works. Nurses see patients every day whose body's immune systems are working or should be working to fight off pathogens. Understanding human immunity helps with proper treatment and assessment, monitoring of patients, implementation of preventative measures, and protection. Nurses and other patients can also be exposed to pathogens very easily. When working with sick patients, it is important to take care of your immune health because a sick nurse cannot properly help a sick patient.